In this lecture, we're going to talk about microbial growth and death. First, let's define what is a living system. Life is able to replicate itself. That's one of the properties of a living system. This excludes things like viruses. While a virus contains the code for all of the viral proteins and the ability to produce more of itself, it lacks the capabilities to do that. It doesn't have the protein synthesis system to enable it to replicate its viral code, to enable it to replicate its DNA. For that, it has to resort to the host cell that it invades to use its systems to produce replicates. In the process, it often kills that host, releasing multiple copies of itself to infect new hosts. So a virus is not alive because it doesn't have the inherent capability of replicating itself. It doesn't have the metabolic network to support that. One of the principles of life is that it exists at a lower state of entropy than its surroundings. Now, from the third law of thermodynamics, all things are supposed to tend towards a higher state of entropy over time. Well, life doesn't do this. Okay, when a cell replicates itself, it's producing more of a lower state of entropy, matter in a lower state of entropy than where it got that matter from its surroundings. That's a key thing about life. It's appearing to violate the third law of thermodynamics. Now, it doesn't really do that because the third law of thermodynamics states that the entropy of the universe, that's both the living system and its environment, has to get higher over time. And indeed, that's what happens. The living cell pulls nutrients from its environment that's in a lower entropic state. So it pulls in nutrients, and it's got to transform its environment by putting metabolic materials back into the environment that are at a higher entropic state than those nutrients it pulled from the environment. In doing that, the entropy of the universe is indeed getting higher, but it's living off the energy and the entropy, the enthalpy and the energy, enthalpy and energy that it is getting from the environment. The cell is able to lower the entropy of the cellular state. So while the universe is indeed following the third law of thermodynamics, the organism is promoting the production of lower and lower entropic states, or more and more low entropic state, at the sacrifice of its environment. So people get confused by this. There's actually a nice section in the book that goes through these arguments. Life also has to have the ability to adapt. And you can, by adaptation, that we say evolve, so it can make a new version of itself that is different subtly or vastly from a previous version. But it also has the ability to adapt to changes in its environment. And a lot of this book is dedicated to that ability to alter how the cell is making decisions relative to changes in its environment. This is something that really wasn't well understood before the 1950s. It was in the 1950s that this idea started to come about. People started finding, hey, there's this protein alkaline phosphatase. This is the classic example. There's this protein alkaline phosphatase that is only produced when E. coli starves for phosphate. That's an example of adaptation to the environment. Long-term evolutionary changes that one type of cell can evolve into a new species ultimately, or you can do gene knock-ins, gene knockouts. Nature can do these same things, and you can alter the genome, you can alter what's being expressed, you can alter the composition, the composition and shape and activity of a protein over time. These are all changes that an organism can undergo over time at its genetic level, but there are also subtler changes at the enzyme level where the enzyme activity can be regulated. And there's an entire chapter of the book dedicated to those kinds of things. So these are the four basic properties of living systems. Now, what enables all this? Well, what enables this is membranes. Okay, if you can't trap the low entropic material and separate it or segregate it from its surroundings, this doesn't work. Okay, so the cell membrane is really the essential component that enables life to exist. Now, it partitions the low entropy components of the living system from its higher entropy environment, but it also creates a barrier that nutrients have to transport across and prevents cell contents from spilling out to the environment. So and then the final component of the membrane is it actually enables what's called metabolic uncoupling. We'll have a separate lecture on that. But this is a key concept that allows cells to evolve. And we'll see that when we get into a lecture on metabolic uncoupling. And it's the fact that the cell can use gradients across the membrane to generate an energy for the cell or generate reducing equivalents for the cell to use interior to the cell that really makes the membrane the key essential component to life. Okay, it's at this level of cellular compartmentalization that distinguishes the two major classes of living systems. And you should all remember from basic biology, we have prokaryotes. These are a single bag of mostly water. If you're a Star Trek fan, you'll remember the Horta from the original Star Trek series. He describes humans as bags of mostly water. Well, that's what a cell is. It's a bag of mostly water. The cell membrane, the lipid bilayer, is the bag. What's inside is mostly water, but it's a lot of biochemicals as well. The DNA, the RNA, the ribosomes, all the enzymes, all the internals that make a cell alive, or all the biochemistry is contained within that lipid bilayer. Now, in a prokaryote, we have a single cell membrane, and the contents are all mixed together. What makes prokaryote different from a eukaryote is a eukaryote has subcellular components. So they're separate membranes inside the cell. Now, it's believed that a eukaryote evolved from prokaryotes, one prokaryote effectively encapsulating another prokaryote. Okay, so chloroplasts and mitochondria actually have their own DNA. They've shared over the eons, they have shared DNA with their host cells such that they're not really freestanding anymore. They require the host to give them some of the materials that they need. They wouldn't be self-standing and self-replicating anymore, but they once were their own living prokaryotic cell. Okay, the nucleus is a little harder to explain how that might have formed, but the nucleus in a eukaryote, that's where the DNA resides, is inside of another membrane material called the nucleus, which separates the DNA and the transcriptional components from, in other words, the production of mRNA from the cytoplasm of the cell. The nucleus pumps RNA out into the cell. There are other things that go on inside the nucleus, one of which is splice variations. That we discuss that in the book. We're not going to go into that in these lectures. But it enabled other things to happen within eukaryotic cells that couldn't as easily happen in prokaryotes. And that actually promotes faster evolution, as well as shrinking the genome. And then there are other smaller components, things like lysosomes, the Golgi apparatus, endoplasmic reticulum, which are also membrane bound. And these are all things that evolved over time. So there's just more compartmentalization in a eukaryotic cell that never existed in a prokaryotic cell. And that enabled other things to happen. It enabled higher life to form. All right, so that's all meant more as a review. Now let's get into how cells actually grow and replicate. Well, we've talked a lot about the entropy and where the cell derives its energy, and we see that in this picture here. Nutrients come into the small molecule pool. These nutrients tend to be small molecules in and of themselves. Things like glucose, for example. Glucose will come in, joins a small molecule pool. Most of those nutrients will go into catabolic reactions, and from that, metabolic wastes are produced, which are exported out of the cell. So we have a higher entropy nutrient coming in, a low entropy nutrient coming in, a higher entropy nutrient going out as a metabolic waste. The net change in the environment outside the cell is to raise its entropy. So, for example, glucose comes in as one molecule, it goes out as 12 molecules, 6 CO2s, 6 waters. Okay, and that means it's less organized, it's a higher entropy, and it's changed the entropy of its environment. Now, a small number of those small molecules get consumed in what's called anabolic metabolism. This is building up the macromolecular structure of the cell. It's replicating the DNA. It's producing extra protein. It's actually allowing the replication of the DNA and then causing the cell to divide. Okay, so some of the nutrients, some of the nutrients go into anabolic metabolism, but making those even lower entropy components requires energy. So in the catabolic reactions, there are reducing equivalents, which are usually in the form of things like NAD, FAD, ferredoxin, where an electron is pulled from one molecule and is transported in a stable form. So these things like NAD, FAD, ferredoxin, NADPH, those kinds of things are critical because they allow oxidation reactions to occur and the electrons to be transported where they can reduce something else. Okay, and then the other thing is we capture energy in the form of high energy phosphates. So this is the ATP ADP cycle that you see here, where ADP is converted back into ATP. The ATP is a high energy phosphate, and it can be used to drive what would be energetically unfavorable reactions when coupled with those reactions to form more macromolecules. Now, none of these biochemical reactions are 100% efficient in trapping that energy, so some of the energy that's released goes out as heat in both catabolic and anabolic metabolism. We can't capture everything, or the cell can't capture everything. Obviously, through evolution, it tries to capture more and more and get more efficient, but not everything can be captured. And this heat, or heat of reaction, essentially, that's not captured in the biochemical molecules, it really becomes the limit in terms of how we can culture cells. At some point, the volume of the reactor gets big enough that you can't control the heat of the cell, the heat that the cells are producing, and you just can't make a bigger culture than that. So any biochemical system generates heat. Just sitting watching this video, your body is metabolizing its breakfast or its lunch, okay, and you've got heat being generated. You have to dissipate that heat, and that limits the size of the body that you can produce. You have to have so much surface area to dissipate that heat. All right, So let's return to how cells grow. Well, microbial cells, I'm going to limit my discussions here to microbial cells. Microbial cells are considered immortal, and they grow by cell division. And whether this is a eukaryotic yeast or a prokaryote, the cell divides, and it divides into two to become two daughter cells. Those two daughter cells are effectively indistinguishable. You don't know which contains the original DNA and which contains the copy of the DNA. Okay, and there's no age associated with that. Both those cells can go on and propagate again. So you have 1 goes to 2, 2 goes to 4, 4 goes to 8, 8 goes to 16. It's a geometric type growth. Now, if we rearrange this equation and take the natural log, we find that the number of generations is equal to the natural log of the number of cells present at any future time t divided by the initial number of cells. So as you can see, this looks like logarithmic growth, or in the inverse form, we can call that exponential growth. Now, the issue is we don't really think, not all the cells are in the same, are replicating themselves at the same time. Okay, and we usually don't think about individual cells. Okay, there's biologists who do think about individual cells. There are synchronizing the cell cycle, that kind of thing occurs. But for the most times, we're thinking about kind of the average cell. What's the average cell doing? And here we really want to relate things back to something we can measure more easily, like time. And it's fairly easy to do that, because if we think about the average cell, okay, and if growth is balanced, in other words, the average cell is replicating on some average cycle, it takes time to accumulate, after a cell division, it takes time to accumulate resources before a cell can divide again. So that time we can characterize as a specific growth rate. Okay, how long does it take for the cell to accumulate, on the average cell, to accumulate enough resources to divide again? Well, we'll call that mu times t, where mu is the specific growth rate of the average cell, and the time is the time it takes between generations for the average cell. So recognize the units on mu are reciprocal time, and we're multiplying mu by time, so we end up with a dimensionless number. So it doesn't really matter now whether we represent the number of cells or we represent the mass of cells, because the average cell will have a given mass. All right, it may grow until it replicates, and then it's smaller than the average, and then it comes back through the average until it gets big enough to divide again. But the average cell has a certain mass, and we call that biomass, which is delineated by X. And if the volume is constant, we can divide X by the volume, and we can determine the concentration of biomass. So this would be mass per unit volume. And since this exponential term is dimensionless, it doesn't really matter how we represent the biomass that's present, either as number, mass, total mass, or concentration. Okay, this exponential applies to all three equally. Now, because this is an exponential, log growth is the same as exponential growth, and you'll hear those terms used interchangeably. They really mean the same thing. They mean that things are growing on an exponential basis, and you can look at it on a log curve, and now it looks linear, or you can describe it as a growing exponential on a linear graph. They're both equivalent. All right. So let's look at the phases of cell growth. We've demonstrated, we've talked about how cells divide, how biomass accumulates, how cells utilize their environment so that they can do that. And but if we think about a batch culture, this biologists think about this, or microbiologists think about this a lot, because a lot of what they do is in batch culture. And you normally start out with fresh media that has no cells in it. You take a population of cells, inoculate the media, and nothing happens for a period of time. Okay? This is called the lag phase. Now, if you're taking the cells from a previously exponentially growing culture that has not gone out of exponential growth, in other words, it's never become nutrient limited, and you inoculate a fresh flask of media that's of the same composition, the same temperature, you will not see a lag phase. Okay? But if the cells were allowed to exhaust the media so that their growth rate has changed slightly, or they've even gotten into stationary phase, then you will see a lag phase, because now the cells were starved for something. They have to reaccumulate that whatever that something is before they can resume growth. So the lag phase is highly variable, and it depends on a lot of things. The log phase, however, is where, or the exponential phase, however we want to call it, it's where there are no nutrient limits on the growth of the cell. The cell is only limited by its ability to replicate itself, or to build enough biomass so that it can replicate itself. So as a function of time, the cells are they're replicating at a constant rate. The mu is constant, and it's the maximum rate at which cells can replicate their cellular contents. It takes time. All chemical reactions take time. It takes time for the cell to accumulate enough material to divide again. That corresponds to a maximum specific growth rate, or mu max. So mu max is a very important parameter. It varies with growth temperature. It varies with the nutrient composition of the broth. The more the cell has to manufacture for itself, the longer mu max takes. The lower the entropy of the nutrients you're providing, the greater the delta, the greater the energy that the organism can derive from those nutrients, the faster mu max will be. So mu max is something that's relatively easy to measure for a given media and a given temperature, and it's pretty much constant as long as you keep the media the same and the temperature the same. That makes it nice. It's a nice constant that we can look at. We determine it once and it holds for all time, kind of thing, as long as we don't change any of the nutrients. Now, mu max, though, depends on there being no nutrient limitations. In other words, the uptake of those nutrients into the cell is not the rate limiting step. Okay, so at some point, because the cells have to keep manipulating their environment to grow, the cells keep growing, they're taking nutrients out of the environment and putting them into themselves, and they're converting nutrients catabolically into metabolic waste products. At some point, they exhaust a nutrient, maybe multiple nutrients, but they exhaust at least one nutrient, and they can't grow anymore. And at that point, we enter what's called the stationary phase. And this is where the cells are unable to replicate themselves. Now, we can think about two types of stationary phases here. There's one type where you're starving for energy resources or catabolic resources. This would be like you've exhausted all the glucose in the culture. All you have left is CO2 and water. And the cell can't do anything with CO2 and water. Okay, there's no way for it to convert it to yet a lower or a higher entropy material. Okay, so at that point, the cell can't derive any new energy from the media. Now, it can convert some of its internal components that it doesn't need at this time and catabolize those. Okay, so it can do that, at least till it runs out of those nutrients. But carbon starvation or energy starvation of a cell is fundamentally different from a byproduct or another nutrient that it might need to manufacture new material. So if we ran out of phosphate in the environment, we still have plenty of phosphate in the cell, and it can recycle that very efficiently, not releasing it back into the environment. If we starve it for extracellular nitrogen, the cell can't grow anymore because it needs more of that to make new biomass, but it can perfectly efficiently cycle that nitrogen back to itself without releasing any to the environment. So these are called mild starvation conditions, as opposed to hard starvation or catabolic starvation conditions. So you can have different types of stationary phase, but think about the stationary phase as it's no growth. We don't have to model it in terms of cell growth, but there's massive changes occurring interior to the cell. There's whole new enzymes and proteins being produced. There's others that are being destroyed and turned off. There's lots of regulatory controls that are going on. Okay, so you have to remember that there's lots of internal change within the cell. The cell actually consumes its own resources. If you've heard people who get stranded on a desert island, they have nothing to eat. It takes you about a month, okay, to starve to death. Well, the first thing that goes is your muscle mass. The next thing that goes is your so you can't move much anymore. The next thing that goes is all the fat in your body, and consume all that. You end up as a skeleton with skin on it. Okay, that's the natural course of starvation, because the body consumes itself to try to keep you alive until you can find nutrients again. All right, there are some cells like Bacillus subtilis. There's some cells that sporulate. Okay, they encapsulate what they need to replicate. They encapsulate that into a proteinaceous spore. Okay, and that spore can survive all kinds of things. There's actually demonstration you can take a spore and expose it into space, as long as you protect it from the radiation in the space. You can expose it to a vacuum, you can expose it to cold conditions, you can bring it back to Earth, and it will open itself back up and regenerate a living cell. Okay, so spores can survive a lot of harsh conditions. Most of our sterilization tests, and we'll talk about sterilization in a minute, but most of the sterilization test organisms are spores. That's what we use to test the ability to sterilize something, because they're very hard to kill. Okay, so the stationary phase, sort of, from gross, there's no new biomass made, there's no new cells made, which means there's no more accumulation of biomass, but there's lots and lots of things happening. That biomass that's there is being transformed into all kinds of other molecules that weren't there during exponential growth. Eventually, however, cells die. Sometimes we try to kill them, like in sterilization. But if they're in starvation, they're in a long-term stationary phase where they're limited to something. At some point, some essential component gets lost, and they can no longer regrow again. At the very least, they can no longer regrow again, or they can't repair themselves. And that's usually in catabolic starvation. The cell loses the ability to repair itself. There's always cellular damage going on, whether that damage is caused by photons of light, or it's caused by chemicals in the solution, or just the physical shaking of the flask that the cells are in. Okay, there's shear force degradation that occurs, and if the membrane finally ruptures, the cell's completely dead because it's spilled its contents into the environment. It's now just part of the environment, and it's at that last phase, when the cell contents spill out, that any cell that's still alive, still hanging on to life in that death phase, can suck up those nutrients, because now the cell was at lower entropy than its environment. It spills lower entropy components into the environment. In theory, those can be used by the cells that remain to regrow or to grow on. Okay, so You can sort of think of cells as little cannibals. They'll eat their neighbors just fine. And that's called cryptic growth. Okay, so we've talked about these 5 stages of cell growth. Now, we'd love to be able to predict what's happening. Okay, if we're, whether we're looking at the progression of a disease, or we're looking at the life of a human, we want to be able to kind of predict. Okay, how is this disease going to progress? Okay, if we're trying to make beer, how long does a vat of beer take to brew? Okay, how long does it take to convert? Once we put an inoculum into grape juice, how long is it before we get wine? And more importantly, how long does it take before that wine turns to vinegar? Okay, so we got to be able to predict these kinds of things, and we need models for all of this. All right, so let's start with the one that most people focus on, and that's the log phase. They want to know how fast are cells replicating. We already know that the concentration of biomass is a function of time, and it's an exponential function of time. And eventually some nutrient becomes growth limiting. Well, this is really what we're interested in here is, we know exponential growth occurs, but we want to know what happens to that growth as we start to get into nutrient limited conditions. Okay, so we're kind of modeling that transition. Well, if we think about it in terms of specific growth rate, the specific growth rate starts, if we're in exponential growth, it's at mu max. So mu would be equal to mu max. So all these models start with exponential growth, and then that somehow changes over time. And over the decades now, multiple models have been proposed for this, but they're all kind of empirical models. And you can see these models here. They involve some sort of constant, Ks, which is normally a fitted parameter. Okay, and a couple of them have more constants in them, like Cs to the lambda here, the Mosier model. Okay, but they're all the same type of shape. They start off at 1, and then they change with the concentration. So they've incorporated the concentration of the limiting component into the model that modifies the specific growth rate from the maximum specific growth rate on that media or on that nutrient. But at some point, that nutrient becomes limiting, and it turns out we can relate that, and the Monod model is the one that most people work with. We can relate Ks to Cs, which is the concentration of the limiting substrate, where Ks is equal to the concentration of the substrate at where mu is equal to 1/2 mu max. Okay, so the Monod model is a really nice, easy one to work with, and if we look at all of these models over here, so as a function of substrate concentration, if the substrate concentration is low, there's no growth. Okay, mu goes to 0. But at some point, the concentration gets higher, and it eventually reaches mu max. So here, this sigmoidal curve, which depends on the concentration, we see that all these models have the same kind of sigmoidal shape, and the Monod model is kind of right in the middle. In fact, if lambda is equal to 1, Mosier's model is equal to the Monod model. Okay, so all of these models predict kind of the same shape to this curve, and it all hinges on what the substrate concentration is. Now, it turns out, and you can see that in a chapter in the book, we can actually relate what Ks means to the ability of the cell to transport nutrients across the cell membrane. Okay, so Ks, for the most part, is membrane related. It's related to the ability of the cell to transport material, to transport a limiting nutrient across the cell membrane. If the nutrient is high in concentration in the environment and low inside the cell, that becomes fairly easy to do. But at some point, the concentration of the nutrient in the environment gets low, and the mechanism the cell has to get that across the membrane against a gradient, for example, changes. It's harder to pump that material into the cell. So at some point, the cell loses the ability to be able to take up fresh nutrient, or the rate of transport, if it was simple diffusion, the rate of transport drops through the cell membrane as the concentration exterior to the cell drops. So all of this is described in a chapter in the book. We're just going to say that we can relate those two here because we want to move on with other growth models. Okay, so Monod is the one that most cells behave pretty well. KS can be fitted in this case if you have growth data that goes through that change into the stationary phase. It's not as clean cut as that because in exponential growth there's usually actually 2 to 2.5 copies of the DNA present. So cell growth may initiate, the cell replication may initiate before or right at the end of the stationary phase and carry through into the stationary phase because the cell has built up enough nutrients to accomplish a round of replication. Okay, so the transition from to mu max, or from mu max down this curve, is not so easy to model, but this seems to work pretty well. All right, so now let's turn to the lag phase. Okay, lag phase has been one that's always challenged people. Typically it's an empirical approximation. We say, okay, the cells, at some time are growing exponentially. If we extrapolate that back to the original biomass of the inoculum, that says there's a lag period because it doesn't come back to t equals 0, and we'll just say the growth is exponential after a lag period. So it's a discontinuity. Okay, there's no growth until t equals t lag, at which point we now have growth. And that's an empirical approximation. You can get by with that. The problem is t lag is not consistent. Okay, it generally does not exist when an exponential culture is inoculated into fresh media, as long as the media and the temperature and everything remain the same. And the lag time increases with the time spent in the stationary phase. So this is why it's so critical to keep your cultures growing in exponential and in exponential phase or log phase growth, to make sure that when you transfer the culture to fresh media, it keeps growing at that same rate. You don't want to have lags because it knocks off all your experiments. It means you have to stay late that day if you're modeling or doing these kinds of experiments. And because it's unpredictable, if you take a stationary phase culture and put it into fresh media in the morning, it may not be ready for you to harvest in the afternoon, or something in between, or before you have to leave. The lab closes for the day. All right. Now, it turns out we can propose a causal model for this. If we assume that there's some growth limiting key component, we'll just call it alpha, that's lost during the stationary phase that must be reaccumulated before growth can resume, then we can say that key component is, we need to know how the cell regenerates that key component. And the easiest assumption to make is that the key component is regenerated in proportion to the specific growth rate. So at mu max, the concentration of the key component, alpha, would be alpha naught. Okay, that would be in balanced growth, that would be how much of the key component is present. And that doesn't change because the cells are growing at their maximum rate. Okay, there's no nutrient limitation, there's no other limitations. It's just the ability to generate that component and other cellular components that allows the cell to replicate. But in the stationary phase, whatever that component alpha is gets lost over time. So we can now look at a model for the lag phase that describes the growth in biomass as exponential growth multiplied by the fraction of key component alpha prime that is present in the inoculum relative to the amount or the concentration that should be there during exponential growth. So if we lost all of it during stationary growth, this ratio would go to zero, and biomass wouldn't grow. Okay, so if we can't lose all of it, if we lose all of it, the cells can't regrow. That's an important point. But we could lose progressive fractions of it. So you can imagine this going from starting at alpha prime over alpha being 1 in exponential growth. Okay, if we have an exponential culture, if it was 5 minutes in the stationary phase, it might be 0.95. If it was an hour in stationary phase, it might be 0.6. Okay, the longer you spend in stationary phase, the lower that ratio becomes, and the longer it's going to take before biomass accumulation resumes. All right. So if you look at this, this is a 3-parameter model. But 2 of those parameters, the initial biomass concentration and mu max, can be determined independently from independent experiments. As we've already said, mu max is the maximum it could grow on fresh media of this type at this temperature. We can measure that over and over again. It's reproducible. We have that number going in. We know what the concentration of biomass was that we inoculated the media with. Okay, we can measure the initial biomass concentration. That's no longer neither of those are fitted parameters. So in reality, there's only a single adjustable parameter, and that's the ratio of alpha prime to alpha naught, or the fraction of key component that the cell possesses when the inoculum is put in to the culture relative to the amount that should be present at exponential growth. That's what alpha prime over alpha is. We've already said that should go to 1 if the inoculum is taken from an exponential phase. Now it turns out if we measure this for E. coli during phosphate starvation, that or I should say, let's back up a minute. I should say that we can relate the log of alpha prime over alpha naught, we can relate that back to T lag. Okay, it's possible to do that. All the mathematics of this are in the textbook. I'm not going to go through them here. But if we can determine what T lag is by a simple back extrapolation of the exponential growth, we can determine what the fraction of that key component was in the inoculum. So if we plot the shift up lag time, T lag, versus time in the stationary phase, and we did this for a mild starvation, phosphate starvation, okay, we can see that T lag increases linearly with time and starvation, but it actually does vary with the growth temperature. Okay, at 42 degrees, it really disappears. Alpha disappears really fast. Okay, at 31 degrees, it actually doesn't disappear very fast. And at other temperatures in between, or so 25 degrees and at 37 degrees, it disappears reasonably fast. It's kind of in between. So it's not a clear-cut function of temperature, but it is linear with time in the starvation phase. Well, what that really means is alpha over alpha naught disappears exponentially, because this is a log term. So if this is going up linearly, alpha prime over alpha naught is going down exponentially. Okay, and we can now calculate From these data, we can calculate alpha over alpha naught as a function of time, and we find out that the rate of loss is a constant. And that's our rate constant for the loss of the key component alpha. All right. Now, let's turn our attention to microbial death. Okay, that's the other side of stationary phase. We've already just said that there's some key component that seems to be lost during the stationary phase that prevents cells from regrowing or slows down their growth until that component gets accumulated. Now, on the other side of starvation, or if we starve long enough, you could imagine that key component may go to zero or may go below one copy. Well, if you get low enough and you can't make more of it, that cell dies, right? So we want to look at that phenomena. Now, before we get there, let's talk about how sterilization works. So we can sterilize, we can actually kill cells, okay? And we do this all the time. We do that to make pharmaceutical products. We do that we have to sterilize the wort before we make beer from it to make sure that we've killed off all the bad bacteria we don't want around, and we can then inoculate the culture with the bacteria that we want. We do the same in wine. We can sterilize the grape juice. You pick it off the vine, it's got all kinds of bugs on it, all kinds of microbes on it, and you got to sterilize that by some mechanism, be it heat, pressure, radiation, or chemicals. Usually not chemicals because we don't want that in the wine or the beer, but certainly by heat, pressure, and radiation. We sterilize that media, and then we put in what we want. And that way we can study one organism at a time. And sterilization is very important. Now, sterilization usually works through what's called the one-hit model. So there's something that is damaged by physical means, by heat, by pressure, it can be damaged by UV or higher energy radiation, or it can be damaged by chemicals. There's something that is damaged in the cell, and it's damaged faster than the cell can repair the damage, and when that happens, the cells start dying. Now, all of these things, in the case of heat, you could imagine as a denaturation of something. Okay, in the case of pressure, nobody quite understands it. Some cells can be killed by pressure, others can't. Radiation, but it's probably a denaturation event of some key enzymes. Radiation's easy to understand. It can actually cut the DNA into pieces. Okay, radiation can damage the cell membrane. There's a lot of things that radiation can do. It damages lots of components. Once that radiation damage accumulates, or it hits the right target. Now, you have to think of radiation as photons, so to speak. There's packets of radiation that are hitting. It's either a high energy electron or like a beta particle, or it could be UV photons that hit or absorbed by something, and it blows that something apart and destroys it. Okay, so that's how radiation is occurring. And if the radiation is constant, it's the cross section it's hitting for the cell is happening at a consistent rate, so it becomes a pseudo first order process. For heat denaturation and pressure denaturation, it's also a pseudo first order process. And the same is true of chemicals. You've got a constant chemical like ethylene oxide around, or bleach around. It's a constant concentration, and as the cells are killed or die off, or the concentration of the chemical stays pretty much constant, and its ability to find those that are still living is now a first order process, or pseudo first order process. So we can model most sterilization processes as first order. Now again, there can be a lag phase. That lag is usually bringing the object being sterilized or the fluid being sterilized up to the sterilizing conditions. It could be spreading the chemical evenly around the material. It could be allowing the radiation to fully penetrate, okay, or the time it takes to turn on the radiation and getting it up to the appropriate beam strength. Okay, so there's a lag period before this pseudo first order process occurs. Often we model that at the same way we modeled the lag phase empirically, and we say that it's first order beyond some period of time. In front of that period of time, we don't worry about, except we say, okay, there's got to be a 5-minute lag before the exponential decay in cell numbers or biomass. We talk more of cell numbers in sterilization because the biomass hasn't gone away. It's just non-viable at that point. It can't regrow. All right. So that's how sterilization works, where we're physically doing something to the cells, physically or chemically. But we also get death in nutrient limitation. And this is a little harder to understand, but we already have a clue. We have a clue that there's some essential component that actually gets lost over time. And this has to be something that's required for its own synthesis. Well, what could that be? Well, think of a ribosome. A ribosome is made of ribosomal protein and ribosomal RNA. Okay, well, if I don't have any ribosomes, I can't make more ribosomal protein. Even if I can produce the mRNA that codes for that ribosomal protein, I can't produce the protein. I can't translate that mRNA without an active ribosome. So if I kill all the ribosomes or destroy all the ribosomes, it's over. I can't do it. I can't make more biomass. That's it. Okay. What about RNA polymerase? Well, if I lose, I needed ribosomes to make RNA polymerase, but I also need the mRNA coding for the ribosome polymerase to make more ribosomal RNA polymerase. And so if I destroy all my copies of RNA polymerase, that's it. I can't recover. So I'm dead. As in terms of being, I may still be existing, okay, but I can't repair myself. I can't reproduce myself anymore. I'm, all intents and purposes, I'm not alive anymore. All right. So there are things like this that we can think of that could actually be viability limiting or essential components for life. Now, we can assume, just based on the model, that all the models we've seen, we can assume that that component is lost some exponential decay, some first order rate. So if I have a lot of that component around, I lose it faster than if I only have a few copies around. You can think of other models here. You could think of an enzymatic degradation. There are other models that we could plug in here, but let's try to keep it simple. It's just lost over time at a constant rate. Now, the probability that a cell contains exactly J of those essential components, starting from an initial copy number of n naught, is given by the binomial distribution. And I've written that out here. Okay, so if I want m components, if m of those components are necessary for the cell to maintain viability, even if it takes 100 years to get out of lag phase, it can get out of lag phase. All right, that it may be a stretch, but at some point that cell could be viable again because it has enough copies that it could reproduce more of what it needs. But we can now describe then the probability that a cell is viable. So we're looking at that loss of n over time from the initial number, and that we can define that as a probability of viability for any given cell. Okay, and we start with all the cells being viable because right at the onset of the stationary phase they are all viable. Okay, they may take time to recover, but they can. At some point though, they lose that viability, and they have to have at least m of those components to remain viable. So we start with 1 and we subtract off all those, the probability of those cells that would have less than m components of those essential components. All right. Now, in the case where m is equal to 1, so if I have 1 ribosome and I have 0, it's a binary choice. I'm either viable or non-viable. At that point, we can solve that equation, and we end up with the number of viable cells divided by the initial number of cells is equal to 1 minus the quantity of 1 minus negative kd over e to the negative kd over t, all to the n naught power. Well, n naught is the initial number of essential components per cell. So I now have a model, and let's see how that works. Well, again, we're looking at E. coli and phosphate starvation. Okay, so it's got plenty of glucose around. We keep transferring the culture so the cells can maintain themselves as long as possible. But at some point, at any of these growth conditions, at some point the cells start dying and they're no longer viable. And we can measure that viability over time. It depends on the culture temperature, and it also depends on what the other nutrients are in the culture media. This was all Selhavi minimal media. These experiments are just done at different times, so it was a glucose minimal media. And you can see that in all these conditions, the cells started dying at some point. They died faster at 42 degrees than they did at 37 degrees, 36 degrees. So the lower the temperature, the longer the cells could survive. You can think about that in terms of maybe there's an Arrhenius equation here. Okay, Kd is a function of temperature. Probably is. We don't quite know yet. But we can now describe this, and we have a fairly simple model because we can measure Kd as the death rate at long time. Okay, so we can measure that. And now if we look at where that comes back, okay, if we extrapolate back to the initial concentration of cells, okay, we get a lag period. Here's where our lag comes in. And we can now look at that as the point at which n over n naught approaches 1. Okay, so we're back extrapolating to where n over n naught approaches 1, and we can solve this equation, and we can solve it for Ts, which is the same as T lag from the empirical model. So that lets us set at this point where the onset of cell death is just beginning. It's kind of a kind of artful way to look at it, but we can now relate Ts is equal to T lag. These terms, we set these two equations equal, and we can solve for the lag time, or we can, if we know the lag time, we can solve for the initial number of components. And if you look, turns out it's a logarithmic relationship. And indeed, if you look at the compilational, now, we've talked about two different components. We've talked about a key component required for nutrient shift up and an essential component required for viability. Well, it makes logical sense. Those would be the same, right? Okay, are they related? Well, the short answer is, unfortunately, no. They weren't related. Okay, the viability limited component seems to change with starvation conditions and the culture temperature, and but the first order rate constants for the loss of the key component and the essential components for viability are statistically different at almost every condition. So you can see here the rate of loss of the key component on nutrient shift up is 3 times at 25 degrees the rate of loss of the essential component was. Okay, it's double the rate or just under double the rate at 31 degrees. They happen to be statistically the same at 37 degrees. Okay, although there's a slight difference there, but it's within the statistical 95% confidence they're the same. But you start getting at 42 degrees, they are indeed different statistically, even though they look close. And if I change from at 37 degrees, if I go from glucose to succinate as the carbon source, they suddenly become different again. Worse, we don't even have a consistent number of initial viability limiting essential components at different growth temperatures and growth conditions. At 37 degrees, we predict 104 for E. coli and phosphate starvation with glucose as the carbon source. 37 degrees, phosphate starvation with succinate as the carbon source. It's 530,000. I don't even know what would be 530,000 copies in an E. coli cell. It boggles my mind. I can't think of anything that would be that high. So it's not like it's one essential component. Okay, that makes it hard to understand it. But in principle, if we could figure out what it was, we could track it during the stationary phase, and we could figure out whether this model really works. All right, so we've looked at everything at this point except for cryptic growth. Cryptic growth is very opportunistic. Okay, it's how fast does the cell membrane lyse open. That depends on the speed of agitation. It depends, because it's usually mechanical breakdown. It depends on what the organism is. It depends on whether the organism's dying off faster than the nutrients can get released. It's very hard to model cryptic growth. It happens at some point. Okay, and it's hard to predict. It's not reproducible. It's very hard to see the rate of cryptic growth, regrowth of the organisms. It's also very hard to track that over time because it's hard to keep cultures alive that long, so to speak. So we don't have models for cryptic growth. We just know that it occurs. But we do have single parameter type models that we can use to predict the onset of the stationary phase, the length of the lag phase, and how long the stationary phase can go on before cell death ensues, and then the rate of cell death. So we have some nice models for all of these things that become very useful, particularly when we're looking at continuous cultures in chemostats or fed batch cultures. It becomes very easy to incorporate these into the growth of the organism, so we have mu as a function of time at all kinds of growth conditions. All right, so that's the end of this lecture. We have some other lectures taken from this book. If you want details on the derivations, they're in the book. I encourage you to buy the book, obviously. And you can see how we arrive at all these models and what the data is behind them and how they relate to the literature. All these references are there in the textbook. Thank you.